Imagine stepping into your garden, and seeing that reddish-brown, powdery stuff on a plant leaf. Looks like rust on old metal, right?
Yeah, exactly.
Most of us would just think, oh, common plant thing, and move on. But what if that simple rust spot hides? Like one of nature's most complex, really cunning, and economically massive biological stories.
That's precisely it.
That's what we're diving into today. This is the deep dive, where we take your sources, pull out the key insights, and give you that shortcut to being, well, genuinely informed.
Mm.
Our
Uh,
mission today: peel back the layers on a very specific, incredibly complex group of fungi, the Urediniomycetes, specifically the ones we call rust fungi from the order Uredinale.
right.
We're drawing insights straight from a key chapter in Introduction to Fungi. And honestly, I think it's going to change how you think about these tiny masters of manipulation.
Absolutely. It's not just about plant disease, though, their economic hit is huge.
All right.
It's really about exploring this hidden world. Biological warfare, basically. Mind-boggling adaptability, even ancient history tied into it.
Wow.
Our goal for you, listening in, is to get not just what rust fungi are, but really why they matter so much
Yeah.
to ecosystems and, crucially, to what ends up on our dinner plates. Think of it as like a masterclass in parasitic strategy.
Okay, strategy. Let's start simple then. What exactly are Urediniomyces? The source calls them a monophyletic group.
Yeah, meaning they all come from one common ancestor.
Right. And there are about 8,000 species. That feels like a lot.
Is a significant chunk, yeah.
Yeah.
And within that big group, the stars today, the Uredinales, the rust fungi, they make up most of them. But 7,000 species.
7,000. Wow.
Yeah. You have others, like microbotryols causing smut diseases, or sporidiales, the red yeasts.
Mm-hmm.
But the rusts, they're the heavy hitters. Definitely the ones with the biggest ecological and economic footprint.
Okay, so if I'm out there maybe looking closely at a plant, how would I know it's a Urediniomycetes? What are the, unique calling cards?
Well, the really defining stuff is happening at the cellular level. It's about how they handle two really critical genetic processes. Karyogamy.
Which is the fusion of nuclei.
Exactly. And meiosis, which is that special cell division for genetic diversity. These don't just happen anywhere. They happen in specific parts of their spore-producing structure, the basidium.
Okay.
Think of it like having two specialized rooms. One room, the probasidium, for the nuclei to fuse. Then another room, the metabasidium, often with cross walls, where meiosis happens and the spores actually pop out, usually sideways.
Interesting. Specialized rooms. And what about their structure, the hyphae?
Yeah, another key thing if you're looking under a microscope, their internal cross walls, the septa, they're simple, just a single pore.
Unlike other fungi.
Right. Many other basidiomycetes have these complex barrel-shaped pores, dolopores, rust stoned. And you also won't find clamp connections, those little bridges you see on hyphae and other groups. These might seem like tiny details, but they're the definitive ID marks.
Okay, that helps clarify the biology side. But the name rust fungi itself, that's pretty visual, isn't it? Why rust?
It's totally literal. It comes straight from the color of their spores.
Sort of reddish-brown powder?
Exactly. They produce these spores in dense little clusters, pustules, often rugged up on the plant. It makes the leaf or stem look like it's, well, rusting, like old metal. It's a very direct visual cue.
And this isn't new, you said. This goes way back.
Oh, way back. We've got archaeological evidence, rusted bits of cereal grains from the Bronze Age.
No way. Bronze Age.
Yep. And think about this. The ancient Romans were so worried about rust destroying their crops, they had a whole festival, the Robigalia, on April 25th, just to appease the rust gods.
A festival for rust. That shows how much they feared it.
Absolutely. Huge economic anxiety. And some think the Robigalia might even be the origin of Rogation Sunday, blessing the crops. Still around in some churches.
Huh. Fascinating connection.
And that economic importance, it hasn't gone away. The whole wheat versus Puccini gramineae, that's the wheat stem rust, that interaction is one of the most studied host pathogen systems ever.
Yeah.
Because it's still a massive deal for food production.
Right. That historical weight is something. But, okay, you mentioned the complex life cycle. Our source really hammers this point, one of the most intricate cycles in nature. How complex are we really talking here?
We're talking just astonishingly complex, like a biological masterpiece or maybe a really elaborate spy novel.
Uh-huh. Okay.
A typical rust fungus goes through 5 different spore stages. 5.
5 distinct types of spores.
Yes. And often, to complete the whole cycle, it needs 2 different plant hosts. And these hosts are usually completely unrelated taxonomically, like not even distant cousins.
Two unrelated plants. How does that even work?
Well, one plant is the principal host. That's where the main phase, often the longest lasting one, happens. The other plant is the alternate host, where a different, often shorter phase occurs.
And this complexity, that's
Nah,
key to their success.
exactly. It's why they're so resilient, so hard to control. It gives them multiple ways to spread, survive different seasons, and crucially, to mix up their genes.
Okay, let's use that classic example you mentioned, Puccinia graminis, black stem rust on wheat, and its alternate host is barberry.
That's the one. Common barberry is the perfect case study.
All right, walk us through the 5 stages for this one. Give us the play-by-play.
Okay, think of it like a relay race. Each spore type is a different runner, specialized for its part of the track.
Got it. Stage 1. Or maybe stage 2, based on the numbering?
Yeah, the numbering is a bit historical, but let's start with the urediniospores. Stage 2. These are often called the repeater spore
Repeater? Why?
because they're produced in these brick-red pustules, the uredinia, on the wheat, the principal host. And their main job is to reinfect more wheat rapidly.
Ah, so they spread the fire quickly.
Precisely. They're single-celled, stalked, dikaryotic, meaning 2 nuclei per cell, which helps them develop fast. Spiny, thick walls for toughness. And one pustule. It can release 50,000 to 400,000 of these spores.
400,000 from one spot.
Yeah. So you get explosive disease buildup.
Yeah.
The really wild part, wind dispersal. These things can travel hundreds or even thousands of miles.
Seriously, thousands.
Yep. Creates the famous Puccinia pathway in North America. They've detected these spores way up high, like 5,000 feet. They're like microscopic biological missiles carried on the wind.
Okay, so those are the rapid spreaders on wheat. What happens when the season changes?
Good question. As things cool down, those red uredinia get replaced by Teliospores. Stage 3, the overwintering spore.
Ah, the tough guys.
Exactly. They look different, black, raised streaks on the stems. They're two-celled, very thick-walled, built to survive winter. And crucially, inside these teliospores, that nuclear fusion karyogamy happens.
The merging of instructions you mentioned.
Right, so they become diploid, then go dormant, waiting out the cold, usually stuck to the old wheat straw. They're the fungus's winter bunker.
Okay, bunker down for winter. Then spring arrives. What wakes them up?
Warmth and moisture. In spring, the teliospores germinate. And this leads to stage 4, the basidiospores, the barberry infector.
Barberry infector. So these ones don't go back to wheat.
Correct. Each cell of the teliospore sends out a little tube, the promycelium, where meiosis occurs. This produces 4 tiny haploid basidiospores, and they get launched, and this is cool, by a surface tension catapult. A tiny water droplet, Buller's drop, forms, expands, and literally flicks the spore off.
Nature's little catapults. Amazing.
Yeah, isn't it? But yeah, key point. These basidiospores cannot infect wheat. Their only target is the alternate host, the barberry bush.
Okay, so they find a barberry leaf, then what?
They infect the barberry, create a haploid mycelium inside the leaf. This then forms these little flask-shaped structures, spermagonia, on the upper leaf surface. This is stage 0.
Stage 0. The numbering is weird.
It is a bit historical,
Huh.
but these spermagonia produce tiny, single-nucleus spermatia, the sexual messenger.
Messengers.
Yeah, they ooze out in this sweet, sugary nectar. It smells quite nice, actually.
A fungus making nectar.
To attract insects. The insects visit, get the sticky spermatia on them, and then fly to another spermagonium on maybe a different barberry leaf. They act as like mash makers.
Transferring the spermatia for fertilization.
Exactly. They transfer spermatia between different mating types because most rusts, like P. graminis, are heteroecyclic. They need 2 compatible types to get together for sex to happen. These spermatia can't start a new infection themselves. Their job is purely sexual recombination, mixing the genes up.
Clever. Using insects as go-betweens. So after fertilization on the barberry.
Right, now we have a dikaryotic mycelium again. This grows inside the barberry leaf and forms structures called aeciospores, usually on the lower leaf surface. This leads to the final spore type, stage 1, orangey yellow, thanks to carotenoids. The bridge back to wheat.
The return journey.
Exactly. These aeciospores are often brightly colored, orange or yellow, thanks to carotenoids. They form in these cup-like structures, the cluster cup stage. Looks like little orange cups erupting from the leaf.
Visually distinct then.
Very. And like the basidiospores, they get shot out forcefully. But here's the switch again. Aeciospores cannot reinfect barberry. Their only job is to get carried by the wind back to the principal, host the wheat, and start the whole urediniospore cycle all over again.
Wow. That is intricate, doesn't even cover it. 5 spores, 2 hosts, wind, insects, catapults. It's an epic journey.
It really is a masterpiece of parasitic evolution.
Given all that complexity, there must still be things we don't fully understand, right? What are the big unanswered questions for scientists studying these fungi?
Oh, absolutely. It's a huge field. Even with all we know, fundamental questions remain.
Yeah.
Like, how exactly does one fungus manage the molecular machinery to successfully parasitize two completely different unrelated plants?
Yeah, the mechanisms must be different for each host, presumably.
You'd think so.
Yeah.
And how did these five distinct spore stages evolve? Was it step by step? Did some get lost along the way in certain species? We see rusts now with fewer stages, these derived life cycles. How does that happen?
And survival, too. What if one host disappears?
Exactly. How do they persist if, say, the alternate host isn't around for a while? Can they just keep repeating the uredinial stage indefinitely? And how do new races pop up and spread so effectively? There's still so much to unravel about their evolution and ecology.
Okay, so we know the spores travel. They have these stages. But the actual invasion, how does a spore, like a urediniospore, physically get inside the plant and start, taking nutrients?
Right, the break-in. It's another sophisticated process. First, germination. They need water, yes, but they also need to overcome their own germination autoinhibitors.
Self-control mechanisms.
Kind of super potent molecules that basically keep the door, germ pore, locked until conditions are really suitable enough water to dilute or degrade the inhibitor.
Smart. Prevents them germinating too early.
Exactly. Then, attachment. It's multi-step. First, just physical sticking hydrophobic forces. Then, as it hydrates, it forms a proper adhesion pad.
Glues itself down.
Pretty much. The fungus releases enzymes, cutinases, esteroses to sort of etch the surface and cement that pad. Then the germ tube itself secretes more glucans, proteins. It's really stuck fast.
Okay, it's stuck. How does it know where to go in? You mentioned something about touch.
Yes, this is fascinating. Thigmotropism,
Yeah.
response to touch or topography.
Yeah.
For the dikaryotic rusts, like those from urediniospores, physical contact is the trigger to form the infection structure, the opressorium.
That was now.
Opressorium. Think of it as a specialized pressing down structure. And the trigger can be tiny, a ridge, just half a micrometer high, like the lip of a stoma, the plant's breathing pore.
Half a micrometer. That's incredibly sensitive.
It is. The fungus feels that tiny ridge, and in complex signals inside involving microtubules, maybe integrin-like molecules, calcium channels, it all tells the fungus, okay, this is the spot, form the opressorium here, and it can happen fast, like within an hour. Precision targeting.
Wow. Okay, a pressorium formed over a stoma.
Right. Then from the opressorium, a thin peg, the penetration hypha, grows down through the stomatal opening. It swells up just inside to form a substomatal vesicle. And from there, hyphae start growing between the plant cells, eventually forming special cells called haustorial mother cells. These coordinate the next step, punching into the plant cells themselves.
Ah, and that leads to the haustorium, right? The nutrient-sucking probe.
Exactly. The haustorium is the business end. It's the rust's specialized organ for siphoning nutrients directly from the living plant cell.
How does it work? How does it steal the food without killing the cell? Initially, at least.
It's incredibly clever.
Yeah.
The haustorium pushes into the plant cell, but it doesn't actually break the plant cell's membrane. The plant membrane just invaginates, wraps around the haustorium.
So it's inside, but still technically separated by a membrane.
Yes. You have the fungal body, its membrane, it's the wall. Then this gap called the extrahaustorial matrix, and finally the modified plant membrane, the extrahaustorial membrane. There's even a seal, like a gasket, called the neckband.
Okay, complex interface. What's the trick for getting nutrients across?
Here's the physiological coup. That modified plant membrane around the haustorium seems to lack the normal proton pumps, AT passes, that would pump nutrients out of the plant cell into that matrix space. The plant can't easily restrict the flow out.
It's disarmed the pump.
Effectively, yeah. Meanwhile, the fungal membrane of the haustorium has increased AT-base activity. It actively pumps protons out, creating a gradient that drives the uptake of sugars and amino acids into the fungus.
So the plant can't stop nutrients leaving, and the fungus actively sucks them in.
Precisely. And one more trick. Rusts secrete enzymes like invertase into that matrix space. This breaks down the plant's main sugar, sucrose, into simpler sugars, glucose and fructose, which the fungus can absorb even more easily. It's total resource capture.
An amazing level of biochemical manipulation. So this reliance, does it mean all rusts have to be parasites? They need that living host?
Yes. Fundamentally, all rusts are obligately biotrophic in nature. They absolutely require a living host plant to complete their life cycle out there in the wild.
Even if scientists can grow some in labs now.
Right. Being able to grow them on artificial media was a huge research breakthrough. It lets us study them more easily. But it doesn't change their basic nature. In the environment, they need that living plan. They're locked into that parasitic relationship.
Okay, so the fungus is a master invader, but plants fight back, right? They're not just passive victims.
Definitely not. Plants have defenses. The most common one against biotrophs like rusts is the hypersensitive response, HR.
What's that involve?
It's basically localized, programmed cell death. The plant detects the attempted invasion and sacrifices the infected cell and maybe a few neighbors. Kills them off quickly.
Creates a dead zone.
Exactly. Creates a dead zone to starve the fungus because the rust needs living cells. It contains the infection. Wall it off.
And this relates to resistance genes.
Yes, it often links to the gene-for-gene concept. And this is fundamental. Think of it like locks and keys. The plant might have a resistance gene, an R gene, the lock. The fungus might have a corresponding gene called an virulence gene, whose protein product acts like the key. Okay. If the specific key protein from the fungus tries to interact with a cell that has the matching lock, the R gene, the plant recognizes it as foreign, as an invader, and triggers that hypersensitive response. Boom. Infection stopped.
So it's recognition.
It's recognition. And we now know these fungal virulence proteins are often secreted by the haustoria. They actually get inside the host plant cell cytoplasm. They're trying to mess with the plant's machinery. But if the plant has the right R gene, it detects that intruder protein and sounds the alarm, the HR.
A molecular arms race, then. Detection and evasion. But rusts are so successful. How do they keep overcoming these defenses? What are their countermoves?
They have incredible genetic flexibility. That's their superpower. Several ways they generate new variations. First, if the alternate host is around the barberry, in our example, they have sexual recombination.
The matchmaking stage.
Right. That shuffles the genetic deck massively. Creates lots of new combinations, potentially leading to new races that can overcome existing R genes in the host population.
Okay, sex helps them adapt. What if the alternate host isn't there? Which happens a lot, right?
It does, but they have other tricks, anastomosis and somatic hybridization. This is wild. If two different rust mycelia are growing on the same leaf, their hyphae can actually fuse together.
Fuse?
Yeah, and they can exchange genetic material. Sometimes even whole nuclei can swap places between the two different mycelia.
So they can trade genes without the whole sexual cycle on the alternate host.
Exactly. It's like a shortcut to new genetic combinations. Studies have shown you can inoculate a plant with just 2 known races, and the spores coming off later can represent like 15 different races just from this hyphal fusion and nuclear swapping. It's a powerful way to generate diversity even without the alternate host. Plus, you always have standard mutation chipping in as well.
So they have multiple routes to variation. That explains the boom and bust cycle we see in farming, doesn't it?
Precisely. You breed a new wheat variety with a great resistance gene. It works wonderfully for a few years, the boom. Then, inevitably, through sex or somatic hybridization or mutation, a new rust race appears that can overcome that resistance, and suddenly, that variety is susceptible again. The bust.
It's a constant battle.
It really is. That's why breeders try to stack multiple resistance genes pyramiding to make it harder for the rust. Or they look for other types of resistance, like partial resistance. Maybe the plant has a thicker wax layer, making it harder for the fungus to find the stomata. Or it resists infection better as it gets older, a dark plant resistance.
And this connects back to why people went after the barberry bushes so hard.
Absolutely. That huge U.S. barberry eradication campaign starting back in 1918 after that massive 1916 epidemic.
It wasn't just about stopping spores from the barberry.
Well, the direct impact was limited because aeciospores don't travel that far compared to urediniospores. But the crucial benefit was slowing down the evolution of new rust races. By removing the alternate host, you remove the main site for sexual recombination. You drastically reduce the fungus's ability to generate novel genetic combinations. It put the brakes on its adaptation.
That makes sense. Slowing the arms race. So Puccinia graminis is the big one. But there are tons of other rusts out there, right? Affecting other plants, doing weird things.
Oh, thousands. And many has evolved variations on that 5-stage life cycle. Some have lost stages which call them demicyclic if they lack the Uredinia, or microcyclic if they lack both Eschia and Uredinia. Just Teliospores and basidiospores, maybe?
Simpler loops.
Sometimes, but still fascinating. Take Puccinia punctiformis, the thistle rust. It's systemic, makes the thistle shoots yellowish. But the amazing thing, the spermagonia produces strong, sweet smell.
The insect attractant again.
Exactly. Fragrance molecules like benzaldehyde. You can literally smell infected thistles from meters away. It's the fungus perfuming the plant to call in its insect matchmakers.
Incredible. Are there even weirder examples?
How about pseudoflowers? Some rusts, like on Arabis species in the mustard family, they manipulate the infected plant to form structures that look like flowers.
Fake flowers.
Yes, and not just messed up versions of the host's own flowers. They often mimic the flowers of completely unrelated plants that grow nearby, like buttercups,
Why?
to attract pollinators. This floral mimicry combines scent, shape, color, even nectar to trick a wide range of insects into visiting and picking up spores. It's like the fungus hijacks the plant's development to build its own deceptive advertisement. Mind-blowing manipulation.
That is truly next-level parasitism. What about coffee rust? You mentioned its impact.
Himalaya vastatrix. Coffee leaf rust, hugely important globally, and has these weird urediniospores, half smooth, half spiny, and it only infects stomata on the bottom of the coffee leaf.
And its history. It changed things, right?
Massively.
Yo.
Found in Ceylon, Sri Lanka now around 1869. People underestimated it. H. Marshall Ward figured out its life cycle by 1882. Brilliant work. But the coffee plantations were already collapsing.
That wiped them out.
Pretty much.
Yeah.
Led directly to Ceylon switching from coffee to tea production. And it's credited with shifting the entire British Empire's drinking habits towards tea.
A fungus changed British tea culture.
That's the story. And it spread globally. Incredible jumps like wind transport from West Africa clear across the Atlantic to Brazil. Controlling it involves fungicides, old copper compounds are still used, and breeding resistant coffee varieties. But coffee's low genetic diversity makes that tough. It's a constant battle.
Wow. What a story. From ancient Rome to modern coffee cups. Okay, so wrapping this up, what are the big takeaways for us listening if we boil down this deep dive on rust fungi?
I think three things really stand out.
Okay, what?
First, just the unparalleled complexity. These fungi are absolute masters of complex life cycles. Multiple hosts, all those intricate spore stages. It's a level of biological strategy that's just astounding.
Right, the 5 stages, the 2 hosts. It's a lot to track.
Second, their role as economic powerhouses, both destructive and, well, influential. Their impact on farming, on ecosystems, it's immense. From ancient fears to shaping modern global markets like coffee. Understanding them is critical for food security.
Yeah, you can't ignore their impact. And third,
Their incredible evolutionary agility. That genetic flexibility, sexual recombination, somatic hybridization, mutation makes them stunningly adaptable. They're always evolving, driving that constant arms race with our crop breeding efforts. They keep us on our toes.
complexity, economic power, and adaptability. Got it.
Thank you for joining us on this deep dive into the fascinating world of rust fungi.
Absolutely. And maybe a final thought for you to chew on. What other hidden biological battles, these microscopic arms races, are happening all around us, shaping our world in ways we don't even realize? And thinking ahead, how might climate change affect these tiny travelers? Will warmer winters or shifting winds change where rusts appear, maybe challenging crops in new areas? Lots to think about. From all of us at the Deep Dive and the Last Minute Lecture team, thanks for listening.